Epigenetic drugs have been developed, I think, for many years. 5-aza-deoxycytidine, a drug, was discovered 50 years ago in Prague, actually, here in Europe. It has been approved for the use in AML patients and MDS patients. HDAC inhibitors, another epigenetic drug, has been approved for the use in CTCL. But the molecular mechanisms, how this drug really work, has not been solved completely. The idea historically was that epigenetic therapy allows to reactivate genes that are epigenetically silenced. And this idea comes from the early studies that tumor suppressor genes are epigenetically silenced. Nowadays, of course, we know that the epigenome is regulating much more. It's regulating enhancer and regulatory sequences almost more than promoter sequences of genes. And these drugs that we are studying here, they have genome-wide effects. That was known. It's not a targeted drug to a specific gene. It's targeting the epigenome. And what the clinical people tell us is that these drugs have a beneficial effect in the treatment of older AML patients, for example, or MDS, where other drugs don't work. And this is sort of where we started, and we asked the question, what can we learn about the molecular mechanisms? And we discovered very surprisingly that these drugs activate repetitive sequences, so-called long terminal repeat sequences, LTR sequences, that are derivatives of human endogenous retroviral elements. Full-length human endogenous retroviral elements have two LTR sequences at the ends, and they transcribe double-stranded RNA, but double-stranded RNAs are detected in the cell, and there is an immune response that triggers these double-stranded RNAs. The LTR sequences that I'm talking about are sort of remaining sequences from older insertions. They only contain the repeat sequences. They have regulatory sequences, but in the normal cell, in almost all normal cells, they are inactive. They are silenced by mechanisms that we don't understand. And now, upon treatment with epigenetic drugs like 5-aza-deoxycytidine or HDAC inhibitors, we start to activate those repeat sequences. So we are activating a promoter and creating a novel transcription start site, TSS, as we call this. And this transcription is then spliced into existing genes, creating what we call TINAT sequences, treatment-induced non-annotated transcription start sites, creating chimeric protein sequences, peptide sequences, truncated proteins, and so on. So almost creating a chaos in the cell, and we believe that this could be the cause of cell death that results from the activation of these repeat sequences.